Okay, hi everybody, and welcome to our first Science Friday talk for this academic year. And we would like to welcome our new COB students and the second year students too. Also, we would like to welcome all the KCU attendees on both campuses and our guest attendees from the Kansas State University, University of Missouri, Kansas City, and University of Texas Medical Branch. So I'm honored today to present to you Dr. Rick Domann. Dr. Domann is a professor of free radical and radiation biology at the in the Department of Radiation Oncology at the University of Iowa. He's the director of the interdisciplinary program in molecular and cellular biology at the University of Iowa too, and hold a secondary appointment at the departments of surgery and pathology. Dr. Domann received his bachelor degree in biology from University of Wisconsin, Platteville, and his PhD in human cancer biology from the University of Wisconsin in Madison, and he finished his postdoctoral studies in molecular oncology from the University of Arizona. He is a world expert on the role of metabolism and oxidative stress on epigenetic and genetic regulation of gene expression and their effects on cancer etiology, progression, and therapy. He has received many NIH grants, including R1 and PO1, and he have have over 153 cancer biology related publications, and he served on many review study sections for NIH, Department of Defense, and Department of Energy. Dr. Domann has received many awards for his contribution to his field, including the Distinguished Scientist Award from the Society of Experimental Biology and Medicine, and he served as the president of the Society for Redox Biology and Medicine in 2018. Dr. Domann had trained numerous number of graduate students, medical resident, MD, PhD students, master students, and his graduates went into different career paths, including PIs, tenure faculty, industry, and teaching. His excellence in mentoring had been recognized many times. To mention a few, as a mentor in excellence from the Radiation Research Society and the Council of Society for Free Radical and Medicine, and outstanding faculty mentor from the Graduate College of University of Iowa. And on personal note, Dr. Domann had taught me 2 classes back when, during the my graduate study in radiation biology and cancer biology, and without him, I really won't even understand what radiation biology is. I was an immunologist who got transplanted into Iowa radiation biology program, but I really enjoy it. He mentored me many times, and I have collaborative work with his lab members. I think we have 2 publications with him, and it's my honor to really have him as our guest. And for today's presentation, he will talk to us about extracellular superoxide dismutase and its role in cancer. Dr. Domann, the virtual room is yours now.
Well, first, I just want to say thank you for that overly kind introduction. And when I sit here and listen to all that stuff, I wonder where I found the time to do all that stuff that you mentioned. So it's an old proverb that says that life can only be understood looking back, but it can only be lived going forward. So there's a paradox that we all face with our productivity and trying to gauge where we are in the scheme of things. Okay, so thank you again, Ihab, for having me here today. And with no further ado, let's get right down to business and discuss the role of extracellular superoxide dismutase in human cancer. Ihab mentioned, I'm a professor of radiation oncology. I'm also in surgery and pathology, so I'm in all these clinical departments, but I have a PhD. So sometimes I feel a bit of a fish out of water, but I believe I have over the years made substantive contributions to all those programs. And I'm delighted today to talk to KCU and the other affiliated Kansas participants that are watching. I heard there are more than 100, so I hope I'm worthy of your time this morning. Let me start by talking about the superoxide radical. This is the one electron reduction product of molecular oxygen. So normally when we respire, we reduce oxygen by 4 electrons to water, which is harmless. But occasionally, the abundance of oxygen and electrons in the mitochondria allows for the 1 electron reduction to superoxide, a radical anion. And it's estimated that about 1% of all the oxygen we consume goes through this 1 electron reduction to superoxide. And 1% might seem like not much, but when you consider that we consume probably on the order of pounds of oxygen per day through our breathing and respiration, 1% can be a considerable amount of this. And so this needs to be taken care of. It's a damaging reactive oxygen species and creates damage to biological molecules. Now, as I just mentioned, it's normally formed during mitochondrial respiration. It can be generated by certain enzymes on purpose in cells, notably the NADPH oxidase family of enzymes. And moreover, because ionizing radiation can liberate aqueous electrons that can then simply adduct to oxygen, it's a product of ionizing radiation. And this sort of explains a little bit about my home department as radiation oncology too. So there are enzymes that work on this superoxide to dismute it or to detoxify it. And it's an enzymatic process through a ping pong mechanism. 2 superoxides enter and out comes 1 molecular oxygen and 1 hydrogen peroxide in the dismutase reaction. And so you can see that you recover 50% of the oxygen that was lost in the previous reaction I showed you. And also you generate another reactive species, not a radical, but certainly a reactive oxygen metabolite called hydrogen peroxide. We usually depict it as H2O2, but another more sort of conceptual way of thinking of this molecule is as HOOH. So, because that's really its geometry and how it exists. At any rate, the SODs are were the first known enzymes that work directly on a free radical to do a detoxification reaction. Now, there are 3 types of superoxide dismutase. They're encoded by 3 different genes. SOD1, 2, and 3 are the names of the genes, and the proteins that they encode are copper zinc SOD, which is a cytosolic version with this activity, SOD2, which is a mitochondrially targeted version of this activity, and SOD3, which encodes extracellular superoxide dismutase, which goes through a lysosomal Golgi secretion process to become an extracellular version of this activity. So this activity exists, in the cytosol, in the mitochondria, and also in outside of the cell in the cellular microenvironment. And so today I'm going to talk about the role of SOD3 and the extracellular microenvironment in the progression of malignant disease. Now, while I'm on this slide, all 3 of the SODs share the activity of dismuting superoxide, and it's important to note that superoxide and nitric oxide, another free radical that's an important biological signal mediator, have a direct relationship that's mediated by SOD, because these 2 radicals, superoxide and nitric oxide, can directly react here in the bottom of this panel, you can see superoxide reacting with nitric oxide at a very fast rate constant to produce this oxidant species, a form of a nitrogenous bleach called peroxynitrite that can further decay into other damaging species. This is damaging in its own right, but more important, this reaction destroys the bioavailability of nitric oxide, which is important in vascular relaxation. And so without SOD, nitric oxide might never get to its intended target to do its biological function. At least without EC-SOD, nitric oxide can traverse several cell diameters, as we know, to mediate its vasorelaxive effects. And notice that this rate constant for this radical-radical termination reaction rivals that of the rate constant of the SOD enzymes. They're both in the 10 to the 9th per mole per second range, so that actually NO competes with superoxide dismutase for superoxide. You can think of it that way. The rate constants are comparable, so in a microenvironment where there's one but not the other, there could be an excess of the third. And let me just show that in an illustration here. Here's a cartoon by Dr. Crapo. Showing an epithelial cell that's got a mitochondria and some cytosol and nucleus, and they're making some superoxide, and they're generating some at the membrane as well. And out here in the extracellular space, we have EC-SOD, which is dismuting this extracellular radical to H2O2, which can be further detoxified to water. This enables nitric oxide from this cell or a neighboring cell to diffuse across this relatively large space to its intended target, a vascular smooth muscle cell. And this could be a vascular smooth muscle cell surrounding a vessel, or it could be surrounding ductwork like mammary gland ductwork or other glandular structure. And so the presence of EC-SOD in this environment keeps superoxide from perturbing NO signaling. But in contrast, if SOD is lost in the extracellular environment or absent or redistributed for some reason, then in the absence of extracellular SOD, the superoxide and the nitric oxide can react. Here, these are the two of the cell origin, but also in inflammatory processes, you can have macrophages and neutrophils undergoing a respiratory burst that make both of these structures, both of these radicals, that can lead to the formation of these this suite or this symphony of highly damaging reactive oxygen species. And many of these species are well known not only as DNA damaging agents, but out here in the extracellular space, they can damage tissue components. They can damage collagen and fibronectin, and they can damage other extracellular structural components of the basement membrane and other connective tissue components. And so that's actually a mechanism of certain human disease processes. And cancer is one of those, and I'll be discussing that in more detail today. So let's see. I'm going to talk about several mouse models of EC-SOD for experimental study, and I'll go through these sort of in order. I'm going to talk about cell transplantation with SOD overexpressing cells, or SOD3 transgenics, SOD3 constitutive knockout mice, and conditional knockout mice, and we'll compare and contrast a little bit. And then I'm going to tell you about gene-doped mice, because we've used these in some fairly recent studies in data that I will show you later in the talk. So first, the first of these is cell transplant with SOD3 overexpressing cells. So in this experiment, what the authors did was transplanted mesenchymal stem cells that were either given PBS or transfected with an empty vector or transfected with an EC-SOD expressing vector. And then they took these mesenchymal stem cells and they injected them intravenously into the tail vein of mice. And then after some cell transplantation and graftment time, they exposed those mice to ionizing radiation, which is known to generate superoxide and those other radicals, as I previously mentioned. And what they noted was that the mice that expressed EC-SOD from this mesenchymal stem cell transplant were highly resistant to a single dose of 9-gray cesium-137 whole body gamma irradiation, compared to mice that were basically untreated or treated with vehicle or treated with empty vector, which showed the normal degree of sensitivity to this level of radiation. So this is exciting because, here right away, cell transplantation with EC-SOD overexpressing cells is a radiation mitigating effect. It has the ability to protect a whole organism from death upon cell transplantation of these MSC cells overexpressing EC-SOD. So that's kind of cool, and not a lot of follow-up has been done on this, but I'd really like to see more of this done and translated to SOD pharmaceuticals as mitigators and other things, for example. So then there is a pulmonary histology and function. There's a pulmonary histology associated with irradiated mice. So this is normal lung tissue, and this is lung tissue from a mouse that's been irradiated with 15 gray of ionizing radiation to the whole body, and then 14 weeks later, and you can see that this mouse that overexpresses EC-SOD, now this is an EC-SOD transgenic mouse that I'm talking about now. I switched models. Remember, I'm going to go through several examples of models. So this is an EC-SOD transgenic mouse. On this side is the transgenic. Here's the wild type, and here's the transgenic. And what you can see is that, again, like before, the SOD3 mouse is resistant to this single dose of ionizing radiation, what would normally have caused a large edema and an inflammatory response and numerous physiological complications was mitigated by systemic overexpression of SOD3 in this mouse transgenic model. Again, to the point that SOD3 is a potent radiation protector. Okay, so now there are two forms. We're going to talk about EC-SOD knockout mouse models next. There are two kinds of mouse models. The first is a constitutive mouse knockout, which is where ES cells are deleted from both alleles, and then mice are raised where through every stage of development and through adulthood, they don't have the gene. So these mice, are constitutively knocked out from SOD3 in every cell in the body of the mouse. And what was first noticed is if these mice were aged out, there was really no phenotype. There was no difference between this constitutive SOD3 knockout and the wild type parent control. And Stu Markland scratched his head and said, what's going on? There must be some way to unmask some biological effect. So they exposed the mice to a known toxicity that would generate superoxide. They put the mice in a 100% oxygen environment, which is highly toxic. You can't stand that for very long. And what they found was that the wild type succumbed within, a mean of about 120 hours or so. But the knockouts were more sensitive, and they died after only 90 hours in 100% oxygen. So the difference was, this difference between the wild type and the knockout, and showing that, this constitutive SOD3 phenotype is not really very strong phenotype. You really have to clobber these mice with a hammer to be able to see any difference from wild type. And so that was a bit of a mystery, although it was partially solved when Dave Harrison did microarrays on the constitutive versus the conditional. And he found that these mice, because they're raised throughout life without SOD3, have upregulated an entire suite of anti-inflammatory cytokines that may be able to compensate for the developmental loss of SOD3. Okay? But let's contrast that with conditional SOD3 mice. These are mice that can be fed tamoxifen, and then upon feeding of tamoxifen, they have a Cre recombinase activated, and then SOD3 is removed from their genome. So these mice can be raised, reared, bred, go through development, and go all the way into adulthood with normal SOD3, and then they can be administered this drug tamoxifen. There's an ER-CRE, an estrogen receptor CRE recombinase chimeric molecule that's activated upon tamoxifen, and that leads to the deletion of SOD3 gene biallelically. So the mice then conditionally lose SOD3 when you give them the tamoxifen. And if you let them grow into adulthood and then you give tamoxifen, you see that, look, here's percent survival, and here's the controls and the wild types and the vehicles, and every other mouse was unaffected in room air. But the SOD3 knockout mice were dead in room air by 10 days. So to me, this illustrates a dramatic point of the difference between a constitutive knockout and a conditional knockout, because what a dramatic difference. You know, the constitutive knockouts could live a normal life and you had to put them in 100% oxygen to tease out a phenotype. Whereas if you allowed them to develop and grow and become dependent, you know, on this is a normal process, that they don't have time to do any compensating, and there's no developmental compensation. And if you pull the rug out from under them, they just die. And that's the way it goes. And by the way, they die of a massive pulmonary edema and basically a large infiltrate into the lung. Okay, the last mouse model I want to discuss before I dive into data is the SOD gene doped mice. So we developed some tools that allowed us to introduce this gene using a gene delivery vehicle. And since it's a secreted protein, as long as we injected it into a professional secretor cell, we could get systemic elevation of extracellular SOD. And so we took an adenovirus, several adenoviruses, an empty one, or one that expresses full-length human EC-SOD, or full-length heparin binding domain deleted SOD. And if you refer to the review paper that I sent out to accompany this lecture, you can see the peptide structure of EC-SOD, and you can see the heparin binding domain, which is highly positively charged region of the protein. That binds heparin in the extracellular space and other negatively charged molecules, and it can be lopped off enzymatically in vivo. And so we just expressed a truncated version of it as well. And so here what you can see is when we took these adenoviruses and gave a single dose into the muscle, it was the thigh muscle of the mouse, and then waited 2 weeks and then took this measurement of plasma EC-SOD activity. You can see there's a baseline. Well, this must be mouse EC-SOD activity in circulation, but the human full length jumps that up about 4-fold. And if you delete off the heparin binding domain, you can see the circulating level is dramatically higher. So we wanted to confirm by Western in this plasma also. So here's a Western blot of the same experiment. In this experiment, we injected low molecular weight heparin before either we did or did before we harvested plasma. And what this does is, because of this heparin binding domain, if you inject heparin to the mouse, you can liberate depots of heparin-bound proteins into circulation. And so this is a way to flush heparin-bound proteins like growth factors and like EC-SOD into the plasma. Here you can see a graphic example of that if you look at these two lanes in the middle, where we didn't add low molecular weight heparin and then take plasma and do a Western blot. You see human SOD here, okay, and you see two bands, which you sort of expect the full length and the heparin-bound deleted mutant. And then, but when you give low molecular weight heparin and then do the measurement, you see a dramatic increase in the amount of protein. So that would be about equivalent to this amount of activity in the protein in the presence of heparin. And then over here, you see when the heparin binding domain is deleted, it doesn't matter if you add heparin or not, because, well, it shouldn't matter. The binding domain is gone and that effect should be eliminated, and it is. And so everything works out. And what the cool part about this is we have different antibodies, so we can measure mouse EC-SOD, which they all have, and then we can measure human EC-SOD, which is the transgenic gene doping that we did. And so everything looks great, and I'm going to come back later in the talk and show you some exciting data that we got once we established this model of EC-SOD overexpression in mice. So first I want to talk about this question, and that is, what is the role of EC-SOD in lung carcinogenesis? We've seen the importance of EC-SOD in the lung and how when you take it away and irradiate animals or give them an inflammatory stimulus, they get pulmonary edema and lung inflammation. And so obviously EC-SOD, which is highly expressed in the normal lung and is important for normal lung function. The question is EC-SOD expression and function altered during lung carcinogenesis? Here's just a little bit of data on the histopathology from the conditional knockout. Here's the control with vehicle and tamoxifen, the normal mice. And here's the conditional knockout, vehicle and tamoxifen. And here's the pulmonary edema that they get when you take away the EC-SOD. So in the human lung, as compared to the mouse lung, some work has been done. And in this nice immunohistochemistry on the left here, you can see that EC-SOD is present around the vessels and around the bronchi or tubules in human lung. Here's the alveoli out here, and here's a little microbronchus here. You can see the bronchial cells inside, and you can see the immunostaining for EC-SOD that lines the basolateral side of here, and also it lines the capillary in both areas where there would be smooth muscles. It's these smooth muscles around the bronchiole that constrict when people get an asthma attack or some kind of a thing, and it's these smooth muscles around the vessel which expand and contract in response to nitric oxide to regulate blood pressure and flow. So the first and interesting early finding came from about the mid-2000s. Seems like forever ago now. Things move so fast, but this finding came from Europe, where just a small panel of normal human adjacent lung was resected in a surgical resection from adenocarcinoma. So they had, like 5 paired samples, and they just simply did a Western blot. And here again, you see the 2 characteristic EC-SOD bands, the one that contains the 8 heparin binding domain and the one that lacks it, and they're both in plasma a little bit, or in tissue here a little bit. But what you can really appreciate is that the amount of protein in the tumor was considerably less than in the normal tissue. And so where did it go? And what happened to it? And where is it? And all those things. And is this functionally relevant or important or anything to human lung cancer? Because this is just 5 sets and 5 paired samples, and it's not huge, and we don't know, but it was intriguing to us. And so we picked up the ball a little while after that. And so now I'm going to show you some of our own data. We wanted to work, sort of apply what had been done in mice to humans, in human carcinoma cell lines, and extend the work that I just showed you, because that wasn't that much. So we took a panel of human cell lines that were all validated and authenticated. This is human airway epithelia. This is human mammary epithelial cells. So these are 2 different normal human cell types from different tissue types, mammary epithelia and airway epithelia. And then here are 3 lung carcinoma cell lines, H1975, and you can read them. I don't have to read them. But you can see that similar to what was seen previously, the 2 of the lung cancer cell lines don't express any protein detectably, and whereas normal airway seems abundant, and HMEC is absent, and we'll come back to that later in the talk. Right now I want to focus on these lung cells. And so to couple this protein expression data, we performed reverse transcriptase PCR to measure RNA. And we weren't doing quantitative PCR back then, but really this is a plus-minus screen in a sense, because these are completely absent. And what we found was a mirrored image in the RT-PCR analysis. Normal lung gave a good signal, and a normal human lung fibroblast, as well as human airway epithelial pure cells did. 1975 up here was strongly positive, and we found that as well. And everywhere that we found negative on protein, we also found negative by RT-PCR. So that was of interest, because it suggests that either the gene is deleted or it's silenced at the transcriptional level, because not only isn't there protein, but there's no RNA in these 2 out of 3 lung cancer cell lines we looked at only at this point. But at least it was a foot in the door. It was a clue to something bigger that might be going on. Now, my lab has studied CpG methylation and DNA methylation for 25 years. And Ehab can tell you all this, Dr. Sarsour can tell you all the specifics about that, and we've even discussed it at length several times and collaborated to that event. But in this experiment, what we did as a sort of a quick peek at the possibility that DNA methylation might be involved in the silencing of an intact gene in these samples was to treat the cells with a 5-aza-deoxycytidine, a DNA methyltransferase inhibitor. So if you grow the cells in this compound for several days, it's not non-toxic, it's toxic to the cells, but you can also cause DNA to become passively demethylated, and genes that are functionally silenced by DNA methylation can become reactivated in the presence of this drug. So, and that's what we saw for EC-SOD. Here you see, 1650, it has no protein and no RNA, and again, no RNA, but if you treated it for 6 days with micromolar of 5-AZDC, we could detect EC-SOD mRNA in that cell. So that was a hint that maybe CpG methylation was playing a role in the regulation of EC-SOD, and this had never been determined before. So we went forward with a more detailed analysis and mapped the CpG methylation state of the 5' proximal promoter of the SOD3 gene, shown here at the bottom. And in 550 base pairs upstream of the promoter, there are about 18 CpGs here. There not about, there are 18. It's a quantum. But we measured the CpG status of these different CpGs by sulfite genomic sequencing. And we found, here's a review of the RNA expression data, and what we found was remarkable. It wasn't unique to this gene. We've seen this on a few other genes, but whenever we see it, it's remarkable. And that is that in every cell line or strain, this is a mortal strain, that expressed abundant and detectable EC-SOD, the promoter was mostly unmethylated, very, very few. And what these rows are is each row of circles represents a cloned molecule from a bisulfite PCR reaction. And so we can discriminate individual CpGs, and if they're methylated, I close the circle, and if they're unmethylated, it's open. So that's the key here. And I'm showing you 5 cloned molecules from the PCR prep from each set. And these were picked at random, so you can go pick 10 or 20, and basically the ratios start to look the same, at least in our experience. And so the remarkable thing was in the cell lines that had lost EC-SOD expression, all of these CpGs across this region were hypermethylated. Notably, there was one that was always almost unmethylated, and that's kind of some interest, CpG number 5, that's right here. I think what was more interesting to us was here is this sort of gray shaded area, which encompasses the area that we later did chromatin accessibility on, and also the area which binds this notable transcriptional activator, SP1, SP3 dimer, that's been shown to play a role in transcriptional activation. So if that site is blocked, apparently there's less expression, or at least that's how we would have interpreted this. So that's nice for these cell lines. The SOD3 gene is aberrantly methylated in lung cancer cell lines that lost expression, but what about in human lung and human lung cancer? So we started out like Canula and Crapo with just a very limited set of only 5 tumors and a normal lung. And this is a Western blot I'm showing you here at the top right. And so measured for Western blotted for EC-SOD protein, we can see that normal lung is abundant, and the 5 tumors are all less than normal lung. The loading was pretty decent in this experiment, so I think that's a real result. And you can see that in the last 2, there was no detectable protein in this exposure, and very, very faint detection, if any, in tumor number 3. At the protein level. So of course, we like to do a vertically integrated analysis. We don't just stop at protein, because this is a CpG methylation is a transcriptional inhibitory event. So we want to look at EC-SOD mRNA levels. So we do that here, and we normalize to 1 for normal lung, which now the labels are down here, okay? Tumor 1 through 5 and normal lung. And mRNA is in this upper panel. So you can see that, tumor 1 had a pretty reasonable level of expression, but the other 4 really didn't at the RNA level. And that's pretty congruent with the protein data. And when we measured by mass spec sequenom analysis, this is a different method for measuring CpG methylation in genomic DNA. We measured these sites 6 through 12 that encompass, so if I back up one slide, they're the shaded area there, okay? And those are the ones we measured here. And in normal lung, and here's the scale, no methylation is bright yellow and highly methylated is bright blue. And what you can see if you read up from 6 through 12 CpGs, they're pretty much unmethylated. And here in this expressing lung tumor, they're pretty much unmethylated. And here in T2, they're starting to creep in some methylation. The squares are darker, they're not quite as yellow. There's a blue one showing up. And by T2, the protein was sort of already beginning to be a little bit lower, but not that much. But as you progress over here, you can begin to see that there's some of these tumors are just some of these tumor samples are highly methylated compared to adjacent normal lung. So this was our first evidence of real CpG methylation, not only in cell lines of lung carcinoma origin, but also in tissue from patient-derived lung tumors. So we wanted to go deeper, and we obtained this set of samples which represent normal human lung and stage 1 through 4 lung carcinoma of a variety of types, okay? And these come as cDNA spotted on a plate from a provider. And I can, if anybody's interested, I can get the info for that. I think it's in one of my papers I published previously. But these arrays, these PCR arrays on normal versus lung cancer tissue are commercially available. And so if you, lay out a couple thousand, you can get a few of these plates. And so we applied our EC-SOD RT-PCR primers to this plate. And we found, as sort of we expected, abundant expression in normal lung. But then what was really interesting was that now we have a lot more than just 5 samples. We've got like 45 over here, and they're divided by stage and type. And you can see across all stages that seems to be progressively trending downward as stage increases. And across all types, adeno and squamous carcinoma of the lung, you see this dramatic diminution in EC-SOD mRNA expression. And since we know this is an epigenetically regulated gene now, we would anticipate that these would be due to CpG methylation as well. Before I broach that, let me take a sip and check the time, okay, and say that we can ameliorate the effects of EC-SOD loss in lung cancer cells by transferring the gene back in. At least in vitro we can. So here are a couple of lung cancer cell lines. Here's A549 and 1650. And we've used our adenovirus constructs, and we put in increasing MLIs, 10, 20, 40, and you can see increasing SOD expression. So these are the cells getting empty. They don't have any protein, and the cells that get the virus get plenty. This one is from a cell lysate. So is this one. This one down here is media collected, and since this is a secreted protein, it should be detectable in the media, and it is, as we hope. And at the very bottom is an activity gel showing that superoxide generated by riboflavin and light can be inactivated and keep the gel from turning blue only when EC-SOD is expressed. So this is a non-quantitative measure of EC-SOD activity. It's a zymogram or in-gel activity assay. So that means that we can sort of engineer these cells to re-express EC-SOD, and maybe that might have an effect on their phenotype. And in fact, it does. When we do that, it results in a less invasive phenotype. And we've quantified that here. And what this is a matrigel invasion assay, where a thin layer of a matrigel, which is a barrier to cell migration, they have to eat their way through this proteinaceous substance and then crawl through a hole in a membrane to be scored. on the backside of the membrane. So the cells are plated on top, and then sometime later, those cells on top are scraped off, and the cells on the bottom are counted. And you can see that compared to add empty, here's just a picture of the membrane. It's kind of a dirty picture, but you can see qualitatively the decrease in cells on the back of the membrane by EC-SOD, and you can see the quantitative decrease in their relative invasion percent compared to control or empty vector cells. So real quick, a model here for the transition part of the talk is that in normal lung, we have SP1 bound and coactivators in a relaxed chromatin structure with an unmethylated promoter. We have high EC-SOD expression, and that keeps ROS down and keeps pro-inflammatory cytokines like NF-kappa B down and keeps heparinase from being expressed and degrading extracellular matrix. I didn't talk to you about this today, but if you're interested, we published on this, and you can go look it up afterwards. In contrast, in lung cancer cells, these CpG sites here in yellow are methylated, the stars, and SP1 can't bind. We didn't show this, but others have shown it subsequent to our work. The chromatin is a condensed state. The histones are hypoacetylated. There's a lot of CpG methylation in the 5' end, and there's no SOD expressed. So high level of extracellular ROS, which can feed back into the cell to affect these pro-inflammatory cytokines and lead to changes in extracellular matrix degrading enzymes. So there's both direct effects of ROS on degrading extracellular matrix, as well as activation of genes that deliberately remodel extracellular matrix. And these two phenomena together, we think, lead to the disruption and destruction of biological tissue barriers that prevent the invasion and spread of cancer. So that the loss of EC-SOD early in malignancy, as we saw in stage 1, would facilitate those cancer cells' invasion through the basement membrane and into the surrounding adventitia. Okay, so now let's see. I have probably 15 minutes left or something like that. And so I want to talk about mammary cells and mammary cancer for a bit. Remember back in the talk earlier, I told you that in normal human mammary epithelial cells, we couldn't detect the RNA, and that's true. We also can't detect it in MCF10A, a normal, non-transformed, non-tumor-forming cell line of mammary. We could detect it readily in lung fibroblasts and readily in airway epithelia, but not in these grown in 2 dimensions. And that's odd, because when our colleague Weisheng Zhang in the pathology department at Wisconsin did immunohistochemistry on normal human mammary tissue, here you can see some ducts. They're sort of adjacent sections. They're not perfectly crossed. So, but these are normal human mammary ducts, and stained with EC-SOD antibody. And you can see intense immunostaining with EC-SOD antibody. And if you do Western blots on normal human mammary epithelia or normal human mammary tissue, you can see abundant expression of EC-SOD, both at the RNA and the protein level. So something happened here with the establishment of this strain. Actually, these are mortal cells, so you can't say that it has to do with immortality. These cells are mortal, and they will senesce eventually. These cells are immortal and won't senesce, but they act very similarly to HMEC. They're contact inhibited. They don't form a tumor in a nude mouse. So, what's going on? What happens between the normal in vivo situation where these cells are happily expressing abundant quantities of EC-SOD, whereas you adapt them to in vitro culture and it seems to just disappear? So to address this question and also get at more of the role of EC-SOD in mammary cancer, my then postdoc, now assistant professor at University of Nebraska Medical Center, Melissa Teo Fitzgerald, adopted a technique of in matrigel 3-dimensional culture. So this is a technique that's been established for a number of years by labs on the West Coast. And what you can do is you can embed single cells into matrigel. So it's like a soft agar-assay, except for matrigel has a lot of RGD-containing proteins and plenty of room for extracellular matrix attachments, so cells can form acini or colonies, and these colonies are polarized, and so they begin to mimic 3-dimensional in vivo structures. And so here is just an F-actin stain showing, the blue is the DAPI nucleus. And here you can see integrin is around the outside. And here you can see beta-catenin is on the apical surface. And the integrin 6 is on the alpha-6 is on the basolateral surface. So these are polarized acinar structures that are in some way mimicking, hopefully, the tubular structure in vivo. These are spherical, of course, grown here. If you grow them long enough and keep feeding them, they do start to form little tubules, but they also just can become larger spheres. And so this is the point where we use these cells. And Melissa found, with the help of my capable RA Matthew Fitzgerald, that as compared to two-dimensional cells, shown here in blue, and an RT-PCR plot that don't express EC-SOD protein, when these cells are expressed for either for 5 days or 10 days at three-dimensional culture, you can see an activation and a detectable level of expression of EC-SOD, pardon the screen, compared to normal breast, which gives robust amplification with a CT value, around 20, compared to you get it up to around 30 in a 10-day three-dimensional culture. But this gene is activated at the transcriptional level by culturing it in a condition where you can get it to grow in 3 dimensions. So here, if you just run these these 4 PCR products out on a gel, you see normal breast is really bright, and you see 2-dimensional 5-day and 10 days, there's no signal. That's this stuff over here. And you see 3D, 5-day and 10-day, this increasing signal, and that would be this plot and this plot. So, this is a conventional way of running out the PCR product on a gel, and this is a way of doing quantitative PCR, which we really adapted and had done ever since back in the sort of early 2000s. And this is more than just at the RNA level. This translates to the protein level. Because in a 2-dimensional HMEC culture stained with anti-EC-SOD in an immunohistochemical protocol, you see 0 protein detectable. But in a 3-dimensional acinus, you see here's the blue are the nuclear DAPI, and you can see EC-SOD, and you can see the bright staining kind of around the apical surface a little bit more, which is exactly what we would have expected. And so here then is the activation of this gene. And this is a reversible process. And if you take the cells here at 3D at 10 days and you put them back at 2 dimensions, 2-dimensional culture, the expression goes away. And this is plotted over here, the relative SOD 3D 10 days, and you can see passage 1, 2, and 3. This reactivated 3D expression of the gene collapses down and is no longer detectable if you put the cells back in 2D. And you can go back and forth, and you can keep doing this. So it's a very plastic and very elastic process. Sorry, I don't know why it's doing that. Okay. What we also looked at was the ability of 5-AZDC methyltransferase inhibitor to affect the process. And in fact, we of course use 2-dimensional cultured cells because it's the 2D cells that don't express. The controls don't really express. But when we gave 5-azacitidine, they gained expression. So just like the cell lines I showed you before in the lung carcinoma realm, we could get 2-dimensional HMEC cells to respond to a DNA methyltransferase inhibitor. So this says to me that this process is regulated by DNA methylation. And I will review the promoter structure briefly with you, the 18 CPGs, et cetera. And so when we applied the bisulfite genomic sequencing to cells in 2D and 3D, the 2D HMEC were completely methylated across the region. But when adapted to grow at 3D in those acini, we saw the promoter became unmethylated in association with this expression of EC-SOD. And since we only looked at 5 cloned molecules representative of 5 cells in a population of millions, we, for the reviewer's benefit, took the total genomic DNA from the population and then used a method called COBRA, or combined bisulfite restriction analysis, to show that in 3-dimensional culture, everything was almost everything became unmethylated or shifted from this band, the cut DNA, to the uncut form. So showing that the vast majority of the population in three-dimensional culture adopts this open and unmethylated chromatin structure. Well, what about breast cancer? Same story as lung. I'll make it brief. Look, we started out with this really limited panel of matched normal and tumor, and you can see that in almost all the tumors, I think it was 7 of 9 in this panel, EC-SOD RNA was less in the tumor than in the adjacent normal surgical resection sample. And when we did immunohistochemistry of these, you can see that in a benign lobule, there's plenty of EC-SOD, but in ductal carcinoma, cometotype, these are necrotic cells here, but here's the viable cells, there's less staining. And there's even less in invasive cancer. And when we purchased one of those breast cancer RNA panels that I told you about before, here's normal, and here's stage 1 through 4 of breast cancer, and you can see a very similar pattern in breast carcinoma, human breast carcinoma, mRNA loss with disease progression as what we saw in the lung carcinoma data. And for a number of these here, numbered 1 through 6, we were able to obtain the corresponding tumor genomic DNA. So it was 1 normal DNA and then 5 tumor DNAs. And we did COBRA analysis, the combined bisulfite restriction on that genomic DNA. And that's unmethylated band is above, and the methylated band is below. And you see here in normal and in tumor number 2, that's this one, you can see most of the sample was unmethylated with some methylated, but tumors 3 through 6 were mostly in the methylated form in all of the genomic DNA that was obtained from those tumors. So when these immunohistochemistries were scored, the point was that in normal breasts, there were no tumors that were negative for EC-SOD, but as early as DCIS, 20% were negative, and by low grade invasive carcinoma, 6 of 7, and this only got worse as the disease progressed. So progressive loss of EC-SOD concomitant with progression of disease. This came out around the time of our paper when another group did a bioinformatic analysis and plotted metastasis-free survival. These aren't our own data versus time and months. And they looked at SOD3 and it was in their data set, and they found that high expressors had a much better survival parameter than those that expressed low. And note, the number that expressed low was 296 compared to the number that expressed high was only 42. It shows you a couple things. One, most breast carcinomas present EC-SOD negative, and if they come in positive, they have a better prognosis. So those are a couple of things you can take from this. And if you go back to the discussion paper that I sent through the program admin prior to this, the review there in the last figure will show a breakdown that we did by the TCGA, the Cancer Genome Atlas, and show that this occurs across many types of breast cancers when subdivided by type. Okay, I think I'm going to skip over that one and show you real quick here at the end this gene dope experiment that we did. So these are data we already looked at, and I don't need to show you again. I showed you this at the very beginning. So these are a gene doped immunocompetent mouse model now. Sorry, not athymic mouse model that have been gene doped. So at day -3, they get the viruses intramuscular, IM injection. And then day 0, they get a tail vein injection of a malignant breast cancer cell line, MDA MB231, that is luciferized. So we can detect this in vivo in a non-invasive imaging assay. And then we do that weekly to monitor disease progression. Okay? And that's done by intraperitoneal injection of luciferin, which is the substrate for firefly luciferase. And so when we give the animal luciferin, there are cells that have luciferase glow, and we can image them in a dark box. So that's the experiment. And here are the results. Immediately after tail vein injection, or 30 minutes after, you can see the empty virus, the EC full length, or the heparin binding domain deletion mutant. These are the gene doping strategy labeled here. And you can see here cells have intravenously, these cells lodge in the lung. So that's where they go. And if you image this 48 hours later, you'd get no signal. And that doesn't mean they're all gone, but it means most of them would be gone. But there's enough viable cells left that are tumor cells that repopulate and then grow out. So let's follow this now at week 2, 4, and 8. And what you can see is in the add empty group, you can see the progression of the tumor, looking from weeks 2, 4, and 8, both from the dorsal and ventral side. And somebody needs to mute themselves. Thank you. And also you can see that EC-SOD inhibits this progression, and whether it has the heparin binding domain or not. And so when we plot this, what you can see is normalized photon flux. You can see here's the empty there across all three. This is just a representative mice, but we had multiple per group. And you can see that the photon flux is dramatically inhibited, or the engraftment of the tumors that express EC-SOD in this assay is dramatically lower, and the survival of the mice that express EC-SOD is greater. So that's a gene doping effect, and it shows that systemic expression of EC-SOD can prevent the progression of cancers in this experimental metastasis model. And then we, to see about human CpG methylation, we went back to this TCGA database, and we pulled out all the human breast cancers in the database at that time, and plotted the degree of CpG methylation at a particular cytosine that we chose in the SP1 binding region versus the level of RNA expression. So this is RNA-seq data versus CpG methylation on this axis. And the blue dots represent normal human mammary tissue, and the red dots represent mammary carcinoma. And what I want you to see here is how the blue dots skew out to the right. That is, that they have high EC-SOD expression, and they're rarely above the 50% line on the y-axis, so they have very low CpG methylation. Contrastingly, the tumor samples it's the rare samples that have the expression. Most of them are, here below 500 units. And also, it's very common that they're on the top half of the chart for methylation. So this is human clinical data from hundreds of human tumor and normal samples, showing that in clinical samples of breast cancer, that this is in fact the case, and it makes EC-SOD a desirable target for epigenetic reactivation. Just in the last minute, I just say about these pharmaceuticals that are now coming online through phase 1-2 clinical trials, which are SOD mimetic drugs. And they have a manganese that's redox active, and they can simulate the activity of EC-SOD. These variants are taken up by cells, so they mimic, and they also have no barrier to the mitochondria, so they're basically present in all the cellular compartments where, I told you about the compartmentalization of SOD1, SOD2, and SOD3 at the beginning, but this drug would be present in all of the compartments where those would be. So, for experimental studies, we can use this, but it becomes difficult mechanistically then to determine what are the effects of extracellular SOD versus cytosolic SOD versus maybe even mitochondrial SOD. So these are very useful anti-inflammatory drugs in clinical trials to ameliorate radiation-induced mucositis in humans. And because they have SOD activity, they're definitely worthy of mention in this talk as therapies that could be aimed at malignant progression. So to summarize and conclude, SOD3 is a target for epigenetic repression in both human lung and breast cancer. We have more recent data published a couple years ago now on pancreas cancer as well, with our colleague, the late James Messer. So this seems to be frequently associated with progression of epithelial cancers, and probably not all cancers, but at least lung, breast, and pancreas so far. It's frequently silenced at the transcriptional level by CpG methylation and less frequently by genetic deletion. Sometimes in lung cancer, both alleles can be deleted, but it's infrequent, and we measured it to respond to a reviewer comment. We found it was present less than 10% of the time, whereas abnormal CpG methylation silencing was present greater than 80% of the time. So loss of SOD3 promotes an invasive phenotype in these tumor types, and SOD replacement may be a useful therapeutic to slow the progression of human cancers. So this is one of the final iterations of my lab at the University of Iowa. This work was funded by the NIH. I have to thank you for your attention and let you know that this famous house in the background of this painting, I think this painting is in the Chicago Art Institute, American Gothic, and it's the Gothic window. And of course, but this gentleman right here wasn't a farmer at all, but he was a dental student at the University of Iowa. This house is in Eldon, Iowa, about 50 miles south of where the University of Iowa is located. And this unhappy woman here, was the dentist's sister, who was also recruited by Grant Wood, the artist who painted this painting. So with that, I'll end and take any questions, see if I can figure out how to end my slideshow and see everybody. I'll keep it up here in case I have to refer to slides. I see people, so there are still people. Nice. Thank you for your attention.
Thank you. Thank you. Thank you, Dr. Domann. And if anyone have a question, you can either post it through the chat or open the mic and ask us the question. You can raise your hands. There is the raise your hand function also if you want to ask and get your microphone unmuted.
Ehab, will you monitor the chat and the hand raising for me so that I don't have to do that part?
That's what I'm doing, yeah. Oh,
okay, excellent. These Zoom or these presentations always go better when there's an assistant. So thank you for hosting and co-hosting, Dr. Sarcer. Appreciate it.
Oh, thank you for coming. Thank you very much. So, any questions? I cannot see the chat. I'll find out. So I have a question. Why still we don't qualify SOD3 as a tumor suppressor gene? So it has all the epigenetic, it has all the, the properties of a tumor suppressor gene. So can you elaborate on that?
Yes, I think it's because it doesn't directly affect proliferation. Most of the bona fide suppressors are genes that take the brakes off of the cell cycle in some way. Now, what I would say is that this gene is a metastasis suppressor, and it doesn't suppress the outgrowth per se of a tumor, of a primary tumor, but its absence in a primary tumor facilitates the spread of the primary tumor. And as we know, patients predominantly die of metastatic disease. Almost all epithelial malignancies terminate with malignant disease. So if we can intervene with malignant spread and perturb this metastatic process, then it's possible we could lengthen these patients' quality of life. So I would say that it's not, that's the reason I think it's not labeled as a bona fide tumor suppressor, because if you delete it and inject those cells into a mouse, they don't form a tumor. So it doesn't prevent, it doesn't perturb or take the brakes off cell proliferation in any way, but rather affects the malignant behavior of those cells once they've grown up.
Oh. So I don't see anyone raising their hands or any questions. So, oh, I can see one.
Maggie.
Maggie. Go ahead, ask your question.
I can hear you, Maggie. Go ahead.
I said, sorry, that was an accident. I accidentally clicked it.
Oh, wow. I would say you have to ask a question now. Those are the rules of the road on Zoom, right? I'm kidding. I'm teasing you. I don't want to put you on the spot.
Dr. Domann, I have a question. Are you hearing me?
Yes, please go ahead.
Is induction of any SOD at the level of transcription, I think I saw you mention AHR is a regulator. Are there any other nuclear receptors PPARs, PXR, CAR that regulate SODs?
That's a very nice question. Thank you. Yeah, AHR seems to do so, and I believe there's an emerging literature on PPAR actually, and also I think that there is a hypoxia-related response. It might be mediated through the AHR because you know, HIF-1 alpha binds the same subunit that that the aryl hydrocarbon receptor binds, right? So they share a subunit that's aryl hydrocarbon receptor nuclear translocator. So I think there's a role for hypoxia and HIF-1 activation in the transcriptional regulation of this gene, which makes sense because, you know, it regulates the byproducts of oxygen metabolism. And after all, where do we get oxygen? That's through oxygenation, and we have a hypoxic response to bring in more. So I think HIF-1, I think there's an emerging PPAR literature, and then you know, AHR. So you could argue whether xenobiotics that generate radicals may also do so. So that's a very nice question. Thank you so much.
Thank you.